and the peripheral nervous system, which, sorry, but this message appeared, and the peripheral nervous system, which is composed by nerves that connect the central nervous system to our skin, internal organs, and limbs. At the cellular level, the nervous system is composed by particular types of cells that are called neurons. Neurons are specialized cells that have the ability to communicate to each other and to other cells by sending chemical signals. In this simple neuronal circuit that you can see here, a sensory neuron receives a signal from the outside of the environment, sends the signal, sorry again to hide the names, send the signal to an intermediate type of neuron called an interneuron. The interneuron sends the signal to a motor neuron, and the motor neuron sends the chemical signals to the muscle for the animal to react. Why am I mentioning this simple circuit? Because I want to emphasize that neurons communicate to each other by sending chemical signals, and these chemical signals are belonging to categories: neurotransmitters and neuromodulators. Neurotransmitters are molecules like, for example, acetylcholine, GABA, and glutamate. Usually they activate ligand-gated ion channels, and I give you the example of the acetylcholine receptor. And these molecules are fast acting, and usually they act locally at the level of the synapses. Neuromodulators, on the other hand, are molecules such as biogenic amines like dopamine and serotonin, neuropeptides, growth hormones, growth factors, and hormones. Usually neuromodulators, they activate G-protein coupler receptors, which are 7 transmembrane receptors that are associated to G-proteins. G-proteins are JTPases that upon the binding of the ligand to the GPCR, they get activated, and then they activate secondary messengers. This leads to a slower acting form of signaling, and also neuromodulators have the ability to act in longer distances than neurotransmitters. So a neuron can send a neuromodulatory signal that will act in cells further down the road. For some reason, I can't advance the slides. What do neuromodulators control? As you can tell from their name, they got their name because usually they play modulatory roles. That means that usually when we have defects in neuromodulators, the animal might not necessarily die, but it will have impaired functions. This is not the rule. There are neuromodulators that play very essential roles. For example, dopamine. If an animal doesn't have dopamine, it will not be able to survive for more than 20 days post-birth. But in general, neuromodulators control a variety of behaviors and emotions. They control locomotion, learning and memory, all our behaviors and emotions, the sleep and wake cycle, how we adapt in a changing environment, and also our metabolism. For that reason, defects in neuromodulator pathways cause severe neurological and metabolic disorders. And here I give you examples of diseases and the relevant neuropeptide or neuromodulator that has been associated with it. For example, anxiety and depression, the neuropeptide Y and serotonin play a role in it. Eating disorders, NPY and opioid peptides play a role in it. In ADHD, NPY and NPS. In addiction, oxytocin and corticotropin releasing factor. In Parkinson's, you must all know that dopamine play an essential role. And in diabetes, which is a metabolic disease, insulin plays the crucial role. Come on. Given that neuromodulators play very important roles from a basic biology point of view, but also from a medical point of view, I'm very interested in studying them. And there are two major questions that I'm interested in answering. One is how are neuromodulators carried in a cell? And the second is how do neuromodulators mediate their effect and regulate neuronal activity and behavior. With that, I would like to give you a brief outline of what you're going to hear from me today. First, I'm going to tell you about the carriers of neuromodulators, which are called dense core vesicles, and a few words about G protein signaling. Then I'm going to talk about a genetic screen that I did in C. elegans to identify molecules affecting neuromodulator signaling. And I'm going to focus on two of the mechanisms that we found through which neuromodulator signaling is regulated through a dense core vesicle biogenesis pathway and a GPCR kinase pathway. Starting, I'm going to talk to you about dense core vesicles and G protein signaling. Just to back up a little bit, I want to briefly tell you that most eukaryotic cells in general have two secretory pathways that they use in order to transfer proteins and lipids to the membrane, either for the release or to be used in the membrane. The first pathway is called the constitutive secretory pathway. It's the pathway that is used by all eukaryotic cells. It's called constitutive because it happens all the time, and it is involved in the transfer of material from the trans-Golgi network to the cell membrane, either for proteins and lipids to compose the membrane or for soluble proteins to be released. The other form of a secretory pathway is called the regulated secretory pathway. The regulated secretory pathway doesn't happen in all eukaryotic cells. It only happens in specialized cells, for example, in neurons, in neuroendocrine cells, endocrine and exocrine cells. And one difference that the regulated secretory pathway has from the constitutive is that it needs extracellular signal for the vesicle to fuse to the membrane. And that extracellular signal, what it creates is an increase in calcium that leads to the fusion. This is the reason why it's called the regulated secretory pathway. The regulated secretory pathway is consisted by two types of vesicles. You must all know the synaptic vesicles that are, you can see them in this EM of a neuromuscular junction. They are these small and numerous vesicles that have a clear, they are clear inside. Synaptic vesicles contain neurotransmitters. Dense core vesicles, on the other hand, they are much larger. They have a dense core, and this is how they took their name, and they're much fewer. And dense core vesicles contain neuromodulators. One difference between dense core vesicles and synaptic vesicles is that synaptic vesicles are exclusively neurons, but dense core vesicles are also found in cells like neuroendocrine cells, endocrine and exocrine cells. For example, the beta cells of the pancreas contain dense core vesicles, and they are similar with the ones that are contained in neurons. One other difference between the synaptic vesicles and the dense core vesicles is that the synaptic vesicles are born locally by recycling in the cell membrane. But the dense core vesicles, on the other hand, they need to be continuously supplied by the TGN through this dense core vesicle biogenesis pathway, as we called it. The dense core vesicle biogenesis pathway starts with the budding of the dense core vesicle at the level of the TGN. And this is where sorting of neuromodulators happen. Some neuromodulators that are called neuropeptides, they get inside the vesicle in a pro-neuropeptide form, in a form that is longer than the one that is the active form. And then inside the vesicle, they follow a process that is called propeptide processing. And that happens after acidification of the vesicle. This acidification leads to the activation of the enzymes that are needed for the processing. And then after processing of the peptides, then the mature dense core vesicle follows a maturation process that involves the removal of unwanted cargo that doesn't need to go to the cell membrane for release. The unwanted cargo usually follows the endosomal and lysosomal route. and the retention of the cargo that needs to remain in the mature dense core vesicle. When the mature dense core vesicle becomes mature, it's then confident for and ready for release. And when the cell receives the signal from the outside, then the vesicle fuses with the cell membrane. One of the problems in the field is that this maturation pathway that I described to you simply is highly unknown. Until very recently, not many proteins were known that play a role in this pathway that regulate the removal and retention of cargoes. And our lab was one of the labs that recently discovered proteins that play a role in this pathway. And I'm going to tell you about some of them in a little bit. After the neuromodulators are released, as I said earlier, they activate G-protein coupled receptors. The G-protein coupled receptors are associated with G proteins. G proteins are heterotrimeric JTPases. They are consisted by the G alpha, G beta, and G gamma subunit. G alpha is the JTPase, and upon binding of the ligand to the GPCR, G alpha exchanges GTP for GTP, and that leads to the activation of G alpha and the G beta and G gamma dimer. And G alpha inhibits or activates neuronal activity. There are 4 different types of G alpha. G alpha Q is an essential one, which plays a role in the activation of neuronal activity, and it's one type that I'm going to mention to you in a bit. Another molecule that plays very important role in this pathway is called GERK. GERKs are kinases for GPCR. GERK stands for GPCR receptor kinases. What GERKs do is upon activation of the GPCR, they get recruited to the cell membrane and to the GPCR, and there they phosphorylate the GPCR, and that leads to their deactivation. Therefore, GERKs are considered negative regulators of GPCR signaling. Although they play major regulators of neuromodulator signaling, their exact role, how exactly they control neuronal activity and behavior, is highly unknown. It's one of the questions that I have, and hopefully today I'm going to give you some answers to this question. To answer all these questions and study neuromodulator signaling, I used C. elegans, the nematode that you must know from Andrea. It's a fascinating model system. I will need half an hour to explain why it is a fascinating model system. But briefly, what I want to emphasize here today is that it has a very simple neuronal system. It's only composed by 300 neurons. The asterisk here has to do with the fact that there are 2 extra neurons that are debatable. The so-called CAN neurons, that many people currently believe that they are not neurons anymore. Therefore, the asterisk. So besides the very simple neuronal system, C. elegans has very powerful genetics, and that allows us to find new genes that are involved in the pathways that we are interested in. And the other reason why I picked C. elegans is because it has very well-defined behaviors that by studying them, I can study the behavior of animals when particular genes are mutated. So I'm going to move on by talking to you about a genetic screen that I did to identify molecules affecting neuromodulator signaling. In the video that you see at the left, oh, I have to leave the, give me 1 second. I can't make it work. Here. In the video that you see at the left, you can see a wild type animal which moves in a bacterial lawn, which is the source of food for C. elegans. The wild type animal creates this sinusoidal wave, as we call it. At the right, you see a video of an animal expressing an activated form of G-alpha-Q. G-alpha-Q, as I told you, is a positive regulator of neuronal activity. Therefore, these animals secrete acetylcholine continuously, and they have this very hyperactive and loopy locomotion. When I say loopy, I mean they form these deep body bends. Since these animals have this very characteristic behavior, I was able to do a genetic screen to try and find mutants that would suppress this behavior, and thinking that these are going to be mutants that regulate neuromodulator signaling. So we did a massive genetic screen to be able to find mutants that play a role in neuromodulator signaling and GQ signaling. We found approximately 110 new genes that today I'm going to talk to you about mutants in two categories. We found mutants in genes that play a role in dense core vesicle biogenesis, and mutants in a new pathway that acts downstream of G-alpha-Q, involves another JTPase called Rho, and regulates the activation of an ion channel called NALCN that I'm going to mention to you in a bit. The first category of mutants are mutants in the dense core vesicle biogenesis pathway. So as I said earlier, wild-type animals moves normally in the sinusoidal wave. The dense core vesicle mutants, when we separated them from the activated GQ, so when they are by themselves, they have this phenotype that we call it unmotivated. I also call them depressed because they really seem like they are depressed, they don't want to do anything. This doesn't mean that they cannot move, they are not paralyzed. When you poke them, when you stimulate them by poking them with a worm pick, as we do here, the animals have the ability to move, but they don't want to move by themselves. Another phenotype that the unmotivated mutants have is that they are slower than wild type, and we have quantified this here in this experiment where we count the number of body bends the animal does per minute. Wild type animals do approximately 50 body bends. Hyper1, which is one of the unmotivated mutants, does approximately half. We could rescue this phenotype by putting the wild type Hyper1 gene back in all neurons, expressing it under a paneuronal promoter, meaning that Hyper1 acts in neurons. And then we could also rescue this phenotype by putting the mouse homologue of Hyper1, meaning that Hyper1 is a conserved gene. Seeing that these mutants, they have an unmotivated and not paralyzed phenotype, we thought that probably this mutant might play a role in neuromodulators and not in neurotransmitters. Therefore, we decided to image neuromodulators inside the animal. We took a neuromodulator called NLP21. It's a neuropeptide. We tagged it to Venus, which is a brighter form of GFP. We expressed it in motor neurons in C. elegans. The motor neurons where we expressed it, they form this action all along the dorsal cord of the animal that we can easily visualize them. The advantage of C. elegans is that it's transparent, so we can easily visualize GFP by using a fluorescent microscope. And here you see the region that we visualize. These dots are dense core vesicles. You can see them here in wild type. These bright dots are dense core vesicles. When we look at what happens at Hyper1 in other unmotivated mutants, we saw this reduction in the fluorescence. The dots were still there, meaning there are still dense core vesicles, but fluorescence was less. This means that is less dense core vesicle cargo contained in these vesicles, which could explain that some of this cargo that regulate locomotion are less, so that's why the animal does not move so fast. When we looked at the synaptic vesicle cargo, by tagging it again to Venus, we didn't see a reduction in the fluorescence, meaning that Hyper1 and the other unmotivated mutants do not control the synaptic vesicle cargo. Therefore, that's why the animal is not paralyzed. We wanted to study Hyper1 further, and when we identified Hyper1 as a mutant, it was a mutant without a name. That Hyper1 is the name that we gave it, and I tell you why we gave it this name in a little bit. Hyper1 had this, yes?
Just a little question. Going back to the slide where you showed the movement, this mutation causes the lack of motivation and no other locomotor defect.
Nothing else. The animal has normal sinusoidal wave when you poke it or when you shine light on it, when you stimulate it, when you starve it, they move normally. They're slower, but they move normally.
Okay, perfect. So
they are not ANC, as we call them. ANC means uncoordinated, and they are not uncoordinated. Their forward, the backward movements are totally fine.
And I assume they are neither mechanosensory defective.
They are not mechanosensory defective, yeah. And they are not chemosensory defective either.
Okay, okay. Wow, interesting. Thank you.
Mm-hmm. So, as I said, hyper1 was a gene with an unknown name. We were interested to understand what it might be doing in dense core vesicle, in neuropeptide signaling. What we saw by blasting is that it contains WD40 domains. These WD40 domains are contained in proteins that usually act as scaffolds and recruit other proteins to form large complexes. So we thought that since hyper1 is such a protein, a good approach might be to do a mass spec and try to find interactors of hyper1. So what we did together with my collaborator, friend Jérôme, we expressed hyper1 tagged to GFP. We expressed the rat homologue in a rat cell line that has dense core vesicles, because it is a cell line that has insulin. It's a cell line from pancreatic beta cells. And we expressed it there, and we did a mass spec. And we were extremely excited and surprised to find as top hits proteins that that belong in this category. They are called VPS 50, 51, 52, and 53. VPS stands for vacuolar protein sorting proteins. And the particular ones that we found, they're proteins that belong in two complexes, the GARP complex and the ERP complex. The GARP complex and the ERP complex are almost identical. The GARP complex is composed by VPS 51, 52, 53, and 54 subunits. The ERP is composed by VPS 51, 52, 53, and 50. The ERP is a recently identified complex. VPS 50 is homolog to VPS 54. And by our mass spec, we found that hyper1 interacts with ERP. Looking at the literature, we saw that in massive mass spec experiments where other labs trying to find interactomes of proteins in HeLa cells and in some other cell types, they also found hyper1 as an interactor of ERP components. The Bonifacino lab also a little bit after our publication found hyper1 as an interactor of both ERP and GARP. So our labs have this different result, but from our findings, we thought that hyper1 is an interactor of ERP specifically, and we thought that if hyper1 is an interactor of ERP, then since hyper1 plays a role in neuromodulator signaling in the worm, then it might be that the other ERP components might also play such a role. So just to mention, I forgot what the ERP and GARP complexes are doing. They are tethering complexes, and what that means, they are complexes that are needed for the transport and fusion of vesicles from the endosome to the trans-Golgi network. This is what the GARP complex is doing. And the ERP complex is needed for the fusion of vesicles that come from the endosome back to the recycling endosome, and from there to the cell membrane to go out of the cell. So these complexes play a role in different, although they are so similar, they play different roles. Going back to the worm, we were interested to see whether the GARP and ERP components might also play a role in the phenotypes that we were observing. So we checked for locomotion and the the fluorescence of the NLP21 Venus, as I showed you earlier. So we checked for all these subunits that you can see here, but I'm going to show you only the results for VPS54, which is a GARP-specific subunit, and VPS50, which is an ERP-specific subunit. When we check the locomotion of VPS54, the animals move normally like wild-type animals, but VPS50, they looked unmotivated, identical to hyper-1s, and they also looked slow, identical to hyper-1s. And the same happened with NLP21. When we saw that, we looked at with imaging NLP21 in VPS50 cells, as you can see, there is a reduction in NLP21 venous fluorescence, which is almost identical to hyper-1, but there is no reduction in VPS54. And this means that GARP is not needed for neuropeptide trafficking, but ERP is needed for neuropeptide trafficking. And this brings us to this model where we believe that we showed that hyper-1 is an interactor of the ERP complex, a new interactor, and somehow they are involved in endosomal recycling, and in an unknown mechanism, endosomal recycling is needed for the biogenesis of dense core vesicle. Somehow endosomal recycling defines the amount of cargo that will be found in mature dense core vesicles. In hyper-1 and ERP mutants, there is less cargo in mature dense core vesicles, and that's why our imaging shows that there is less GFP in these vesicles. We were interested to figuring out how this might be happening, and it was difficult to do that in the worm. That's why we decided to betray the worm, sadly, and use a cell line, a mammalian cell line that is called insulinoma cells. It's a tumor derived from beta cells of the pancreas. The reason why we used those is because they contain dense core vesicles. They are used as a model system for studying insulin secretion, because I don't remember if I told you earlier that dense core vesicles contain insulin in the beta cells of the pancreas, and they are very easy to perform biochemistry and cell biology experiments. So the first thing that I did is I created, using the CRISPR technology, and a knockout line for hyper-1. I deleted the hyper-1 gene. I introduced it back in the cells by adding the wild type gene back in the cells to create a rescue cell line. And the first experiment that I did is that I checked for secreted insulin. My rationale was, if the hyper-1 knockout cells have less insulin because they have an impairment in dense core vesicle biogenesis, then they will secrete less insulin. And indeed, that was the case. The way that we activate insulin release in these cells is by growing them in the presence of high glucose and high potassium. High glucose and high potassium, what they do is that they increase calcium inside the cells, and that leads to the fusion of the vesicles. And then we can take the media where the cells are grow and measure by ELISA the amount of insulin. As you can see, in stimulation conditions, wild type cells secrete insulin. Hyper-1 knockout cells secrete much less insulin, and we could rescue that when we reintroduce the wild type gene back in the cells. One way that this reduction could be caused is if the hyper-1 knockout cells have less insulin, because they make less insulin. So we measured the amount of insulin that is contained in these cells, and we didn't see that these cells contain less insulin, meaning that indeed there is some problem with the biogenesis of insulin of the dense core vesicles, and not with the making of insulin per se. And this result has given me the idea that if I make mice that have a defect, or they have a deleted hyper-1 gene, these mice might be diabetic, and also have given me the idea that one of the causes of type 2 diabetes might be patients that have defects in APER1 genes and in the other genes of the ERP complex. So in the future, this is something that I'm interested in figuring out. One way that APER1 knockout cells could have less insulin, could be secreting less insulin, is by making less dense core vesicles or morphologically abnormal dense core vesicles. So we did EMs to see the morphology of the dense core vesicles in the APER1 knockout cells, and we didn't see any defect, meaning that APER1 knockout cells don't have morphologically abnormal DCVs. This means that the dense core vesicle is normally budded off from the TGN in APER1 knockout cells. And therefore, we thought that probably APER1 is acting somewhere downstream of the budding of the DCV during the maturation of the DCV. And one way to see if that was the case was to check what happens in the cell, in the APER1 knockout cells, what happens to dense core vesicle cargo that are either retained or removed during maturation. One of the cargos that is retained in the dense core vesicle is insulin, as I said earlier. And one of the cargos that is removed is the cargo called CPD. CPD stands for carboxypeptidase D. Is a cargo that is needed in the immature dense core vesicle, but is removed because it's not needed in the mature dense core vesicle. So what we did is we did immunofluorescent experiments where we observed where is insulin distributed in wild type APER1 knockout cells and in APER1 knockout cells that express the wild type APER1 gene. As you can see, in this experiment here, we have co-stained wild type cells with a marker that labels the trans-Golgi network. Just to tell you that the mature dense core vesicles are found in the region around the trans-Golgi network. The mature vesicles, on the other hand, they are all down the cell periphery. And you can see that with this insulin staining here. All these little dots that you see are mature dense core vesicles that contain insulin, and they are waiting for the signal to release their content. In APER1 knockout cells, very interestingly, we saw that the mature dense core vesicles in the cell periphery, they contain much less cargo, which is something that we quantified here, where we measured how much cytoplasmic insulin exists in wild type versus APER1 knockout cells versus the background. And we saw that they contain much less insulin, and that could explain why the APER1 knockout cells secrete less insulin, because they contain much less insulin in the mature vesicles. And we also observe that there is something like an accumulation of insulin in the region where the mature dense core vesicles are, something that again you can see here. And we could rescue this phenotype by reintroducing the wild type APER1 gene. This means that it's indeed because of the loss of APER1. So this experiment tells us that APER1 indeed plays some role in the maturation, and it is needed for the retention of insulin in the mature dense core vesicle. Then we moved on to check what happens with the removal of the cargo called CPD. Jerome stained wild type APER1 knockout cells and cells expressing the APER1 wild type gene with TGN38, which is a marker for the trans-Golgi network, and CPD. As I told you, CPD in wild type cells remains in immature dense core vesicles, which are around the trans-Golgi network. You can see it here. But surprisingly, in APER1 knockout cells, we observe that CPD appears in vesicles that are down the cell periphery, and something that doesn't happen in wild type cells. Again, we could rescue that. We didn't know what these vesicles are, but we thought that probably they are mature dense core vesicles. To prove that, we co-stained wild type and hyper-1 knockout cells with insulin and CPD. Again, in wild type cells, insulin goes to the mature dense core vesicles. CPD remains in the immature dense core vesicles around the trans-Golgi network. In hyper-1 knockout cells, insulin is much less in the cell periphery. And CPD appears in these vesicles in the cell periphery, and some of these vesicles colocalize with insulin, meaning that indeed CPD remains in mature dense core vesicles in hyper-1 knockout cells. And this brings us to our final model, where I hope I have convinced you that hyper-1 is needed for the removal and retention of cargo during the maturation process. My technician and friend Michael Crawford has done an amazing work during this last month to prove that the ERP complex is also needed in exactly the same way like hyper-1. By we knocked down the ERP specific component VPS50, and we saw that cells that contain this VPS50 knockout, they have identical defects like hyper-1. This means that hyper-1 and the ERP complex are needed for the maturation of dense core vesicles during dense core vesicle biogenesis. The mechanism still remains unknown, and it's something that I want to investigate in the future. One very interesting paper that came out recently, and I want to point out to you guys, is to you folks. We are not allowed to you guys anymore when women are involved in the presentation. It's a paper in molecular psychiatry, which showed that twins which had psychiatric defects, they showed downregulation of the gene hyper-1. So hyper-1, it was the first time that is shown to be involved in humans in a case of a neurological disorder. And I'm hopeful in the future that I will be able to reveal more cases of humans with neurological disorders that will have mutations in either hyper-1 or in components of the ERP complex. So I'm gonna move on now to the last part of my my talk, which involves mutants that I found that are involved in this other pathway that doesn't have to do with dense core vesicle biogenesis, and it's a pathway involving a GPCR kinase. And as I mentioned earlier, we did this screen to find mutants that that suppress the loopy locomotion and the hyperactivity of activated GQ. One category of mutants that we found are mutants in the channel called NALCN channel in mammalian system. It's called NCA channel in the worm. Why is this channel a very interesting channel? Is because it is regulating the resting membrane potential and neuronal excitability of neurons. It's needed for proper synaptic function. And very interestingly, the last years, several recessive and dominant mutations in the NALCN gene have been directly linked to severe neurological disorders in infants that are born with severe defects, seizures, intellectual disability. Very often, these are cases of kids that die very early on after birth. So for that reason, the NALCN gene has been extensively studied during the last years. It's a sodium leak channel. And in the worm, what it is doing, and this has been shown by Mei Zhen's lab a few years ago, is that it is involved, it is needed to sustain the locomotion of the worm. As you saw earlier, wild type animals do these sinusoidal waves where they can keep on going forever. But mutants in the NCA channel, they cannot sustain locomotion. They do a couple of body bends and then they stop. and we call that fainting. Therefore, mutants in the NCA channel, they're called fainters. One other mutant that I found in the screen that had similar, it was similar in the way that it was suppressing activated GQ, similar to the NCA mutant, is a mutant in the GPCR kinase GERK2. As I said earlier, GPCR kinases are kinases. They have a kinase domain and they phosphorylate GPCRs to terminate GPCR signaling. GERK2 was known to play a role in chemosensation, but it was not known to play a role in locomotion. Therefore, that was very interesting to me that it seemed to suppress activated GQ. I saw that it not only suppresses the loopy locomotion, but it also suppresses the hyperactivity. As you can see, GQ mutants are hyperactive. They move faster than wild-type worms, and GERK2 suppresses that. Interestingly, when I separated the GERK2 mutants from the GERK2 mutants cannot swim. They create this kind of their bodies becomes very rigid and they don't have the ability to swim. This behavior was a behavior that was also described for the NCA channel. So I thought that probably GERK2 somehow regulates the NCA channel. But GERK2 mutants are not fainters. But I thought that it might be that because GERK2 is a GPCR kinase, it might modulate GPCR signaling and it might modulate the NALCN channel. So it might be, as we call it, a partial fainter, something that I might not be able to see it with the simple assay that I'm using. So what I decided to do is make a double mutant and put GERK2 in the background of a sensitized mutant, as we call it. And I chose NLF1. NLF1 is a component that is needed for the NCA channel to act, but it's not in the worm, in particular, it's not essential. So NLF1 mutants are partial fainters, as we call it. They are not as strong fainters as the NCA mutants. And this is something that I have quantified here. But when I combined the NLF1 mutant with the GERK2 mutants, GERK2 became strong fainters, meaning that indeed somehow GERK2 regulates the NCA channel. And knowing that GERK2s are phosphorylating and deactivating GPCRs, I was interested in finding which GPCR this might be. So I looked into the literature and I found this paper from Michael Kohler's lab from 2004, where he had shown that the D2-like, dopamine 2-like dopamine receptor DOP3 negatively regulates C. elegans locomotion by activating GO, which is a form of G protein that negatively regulates neuronal activity in C. elegans and locomotion. So I thought that what if GERK2 acts against DOP3, phosphorylate DOP3 to inactivate it and therefore allows for locomotion. In this case, in GERK2 mutants, DOP3 will be all the time active and locomotion will be impaired. Therefore, GERK2 mutants will be slow, which they are. In order to investigate my hypothesis, I made double mutants between GERK2 and DOP3. I expected that if indeed GERK2 acts on DOP3, then DOP3 mutants will suppress, as we call it, the GERK2 phenotype, because DOP3 will not exist anymore to slow the locomotion. And indeed, that's what happened. DOP3 mutants fully suppress the slowness of GERK2 mutants. Also, they suppress the swimming defect. As you can see it here, GERK2 DOP3 mutants swim normally. And also they suppress the fainting defect of GERK2 NLF1 mutants. GERK2 NLF1 DOP3 triple mutants are not strong fainters anymore. They behave like NLF1s. And then I wanted to see if these animals, the GERK2 mutants, have a defect in the synaptic neuromuscular junction. So I collaborated with Jihang Bei, who is a great friend and collaborator at Fred Hutch, and Yongming Dong, who is a staff scientist in his lab, did these recordings. Here he recorded from the neuromuscular junction of the worm. And as you can see, the amplitude of the evoked postsynaptic currents in GERK2 mutants are reduced, and this phenotype can be rescued in the double mutant GERK2 DOP3, meaning that indeed GERK2 acts against DOP3 to mediate this kind of locomotion signaling. To figure out where GERK2 might be acting to mediate this phenomenon, at first I thought it must be acting in motor neurons since it affects the neuromuscular junction, because the motor neurons are the ones who give a signal to the muscle. So I first expressed GERK2 in motor neurons to see if this could rescue the swimming and locomotion defects, but I didn't observe a rescue. Then I expressed it in interneurons and sensory neurons, which are neurons upstream of the motor neurons. And I could see that only when I expressed GERK2 in the interneurons, I could fully rescue the crawling, swimming, and fainting defects. So somehow GERK2 acts in interneurons to mediate this effect. I collaborated with Laura Pereira, who is an amazing scientist, I mean, she still is an amazing scientist, but she's not in the Oliver Hobart lab anymore. She moved on. She was a postdoc at the Hobart lab, and she can identify where a protein is expressed. So I sent her animals that had tagged GERK2 with RFP and a marker that expresses GFP, YFP in cholinergic neurons, and she found that GERK2 is expressed in all these interneurons that you can see here. You don't care about the names, but it's a bunch of interneurons that are found in the head of the worm. So indeed, GERK2 is expressed in these neurons and functions in these neurons to regulate this behavior. And then I did the same for DOP3, and I proved that DOP3 is also expressed in the same type of neurons. And when I expressed it in interneurons in a GERK2 DOP3 mutant, I could reverse the DOP3 rescue. This means that indeed GERK2 and DOP3 act in interneurons to mediate this effect. My favorite model that I currently have is that GERK2 negatively regulates the DOP3 receptor, which is, as I said, a D2-like receptor that is associated to GO. GO negatively regulates GQ, which, as I've seen I mentioned briefly earlier, activates the GTPase rho, which in an unknown mechanism activates NALCN channel and sustained locomotion. This is the model that I'm currently investigating. Very interestingly, a mouse lab was able to show using electrophysiology that in mouse brain, a D2-like receptor negatively regulates NALCN channel, which means that the pathway that identified first in the worm is a conserved pathway that was proven to exist in mammalian systems as well. How much time do we have? Andrea, how much time do we have?
By the clock, we have like 4 minutes. Okay,
then I'm gonna wrap this up because this is I had a very little story to say, but it's taking too long. So in this last story, I hope that I convince you that GERK2 acts on DOP3, which negatively regulates the function of the motor neurons by negatively regulating crawling, swimming, and the NALCN channel. In another study that I haven't published yet, and I'm currently investigated, I have found that GERK2 also acts in the sensory neurons, and that regulates the aroused state of the animal. How this means how active, let's say, the animal is. It regulates a form of a locomotion sleep that C. elegans has. I don't have time to talk to you today about that. Hopefully in another meeting in person in Valparaiso soon. With that, I would like to thank you all for your attention, and especially to thank Andrea for inviting me over. Special thanks to the Elion Lab, especially to Michael Crawford, who is the guy you see here, an amazing technician who works with me. Previous members, Jerome Caten, a friend and collaborator. Special thanks to the Bay Lab and to Jihang Bay, who is my collaborator and friend, to the Raisin Lab for helping me with studying the sleep behavior that I didn't have the time to mention, Hobart Lab, and especially Laura Pereira for doing all the imaging, and the Hopkins Lab for helping me with cell cultures and things like that. And with that, I will wrap it up, and I'm happy to take questions if you have any.
Thank you so much, Irini. It's so fascinating. I have many questions, but I'm gonna let other people ask first.
Sure.
So the session is open for questions. Let me see. Okay, so maybe
you can raise your hand. If people have any question, they can raise the hands.
But maybe I should start. No,
Pablo.
Pablo is there.
Well, thank you. Thank you for the wonderful, results that you are presenting. I was wondering if in this dopamine receptor in C. elegans, have you found any indications that the GRIK2 interaction might differentially impact the signaling towards the canonical G protein pathway versus beta-arrestin homolog? Something like, I'm thinking about functional selectivity or biased agonism.
Thank you very much, Pablo. So it's very interesting, but beta-arrestin in the worm, we haven't seen it playing any role yet, which is a very, very interesting phenomenon, and something that I'm very interested in studying further, because there is a beta-arrestin homolog, and I haven't managed to see it having any phenotype in the studies that I'm doing. So it seems that GERK2 acts in a beta-arrestin independent way, which is a difference with mammalian systems. So for that reason, I cannot really address your question, since beta-arrestin is not acting. Thank you, though, for the question. Yeah.
So I'm gonna go ahead with my list of questions then.
Go ahead, Andrea.
I wonder if I'm in the first part of the talk, right? The IPER1, do you think that's a good model for studying depression? That's one of my first questions.
I very much think so. Yes, I think that all our unmotivated mutants that we have, and we have a bunch of them. I mean, I was able to only talk about IPER1 and the ERP complex. We have many more. And I believe that all of them, some of them have already been associated with anxiety, with cases of anxiety. Some of them have been associated with cases of diabetes. I'm very sure, I mean, I cannot say that I'm very sure, but I'm very hopeful that in the future we will find patients with defects in IPER1 in ERP that have cases of depression, ADHD, anxiety disorder, stress disorder, all sorts of neurological disorders.
So that takes me to the other part. What do you think makes a worm model qualify as a depression model? Because a lot of people are interested in that. So I was wondering if you could illuminate me there.
It's very hard to answer this question because you can never ask the worm if it feels depressed. But if you look at the worms, and I can send you some of the unmotivated worms that you have, you can tell it from a human point of view that this animal must feel depressed, because it's really not wanting to do anything by itself without stimulation. So, of course, somebody can argue that this is how I see it, but given that the only way you can figure out if an animal is depressed, since you cannot ask it how it feels, will be by seeing if it wants to do tasks that it will do in normal times, right? And by looking
for example, finding food, do you think that, do they have a problem in finding the right, the food source?
I haven't tried doing that, actually. I haven't investigated how much these animals, because I'm focused on their locomotion phenotypes. I have tested whether they can sense food, like if you put the food in front of their nose, they can sense it and they back up. But I don't know how easily they will be able to detect food, look for food, and things like that. So meaning how motivated they are. I think that they have a problem with being motivated, and that's a classic dopamine defect, because dopamine is a neuromodulator that is giving the feeling of motivation, a reward. So I think that the worms have a problem with being motivated, and that is a form of depression in the sense.
So there is Diego who has a question, but after that, I would like for you to tell me whether males can be motivated to have sex with the hermaphrodites. But let's leave Diego to have a chance.
Thank you very much, Andrea. Irini, a nice talk. It was very interesting. My question is about motivation and dopamine, but thinking about error prediction, because if those C. elegans are, I wonder if you have studied if they can learn associatively the mutants like the wild types, and also I wonder if you have studied if there is something in their signaling of error prediction that changes in your mutants. Does it make sense, my question? I don't know if English
Your
is good.
question makes excellent sense, and it's a great question, and we haven't studied any of these things because of funding issues. We have focused on the mechanism of dense core vesicle biogenesis because this is what our funding is funding right now. So we have put all our effort in figuring out what's happening at the level of dense core vesicle biogenesis. So we hadn't had the chance to investigate behaviorally whether these animals have problems, as you said, with associated learning and stuff like that. We haven't done any such assays, but these are very interesting questions that I'm very curious to investigate in the future. Thank you, Diego, for asking.
Thank you very much.
And Andrea, to answer your question, so males that are unmotivated, they can mate. You have to put much more on the plate, so they really have a defect. They are not unable to do it, but they are suffering. They need more. You need to put many more males than in normal mating plates.
So that adds to the point, right? I don't know whether that's the only criteria, but that also adds. And I wonder, you remember that sometimes Marty told us that older males are, even when they are mechanosensory defective, they are extremely motivated to mate. So when they are old, these are not young animals, right? But when they are old, they sort of overcome the problem that they have with mechanosensitivity, and they go out and mate, and they are very active at doing that. So I wonder if you can test this type of motivation in older individuals.
I have not done any such things. All these behavioral, all these experiments that you guys are proposing and Diego proposed are excellent experiments, but I have focused so much, we have focused so much on the mechanism of function that we have put the behavioral aspects aside. One other reason why we have done that is because we haven't been able to find which neuromodulator or which neuropeptide is responsible for these behaviors. We have not even been able to figure out which neuronal type is responsible for this behavior. We have come to the conclusion from a series of experiments that it's not only one neuronal type, it's not only one neuromodulator, that it's a combination of them. That's why it's kind of harder to address as a question. But generally, I strongly believe that these are mutants that can be used as a model for depression and studying further behaviorally what exactly is happening and what is the mechanism behind what's going wrong. Because I strongly believe that such mutants in the future will be found in humans with neurological defects.
There is Chayu who wants to ask a question. Go ahead, my friend. There she is.
So thank you for the wonderful presentation. I have a question for the first part also, and related to your just previous answer. Is it known? So do you think that hyper-1 is involved in all release? So the dense core vesicles for any neuromodulator, any cell type, have people staying for them, in different cell types?
Question. Very, very good question. So far, I have seen it being required in 2 different systems, in C. elegans and in pancreatic beta cells. But there are a student in our lab is studying 2 different neuropeptides that are present in a cell type called ASI neurons in the worm. The ASI neurons secrete approximately 20 neuropeptides that are localized in different areas in the cells, which means that they are contained in different dense core vesicles.